Today, we're diving into a really exciting breakthrough in the fight against superbugs. We're going to break down how scientists figured out the secret weapon of one of the world's most dangerous bacteria and, more importantly, how they might be able to disarm it. So imagine this. One of the world's deadliest bacteria has this secret weapon, a nearly impenetrable shield it builds around itself. For years, this shield has made it incredibly tough to treat. But now, it looks like researchers have finally found a chink in its armor.
So first up, let's meet the superbug at the heart of this whole story.
Okay, so Staphylococcus aureus, or just S. aureus for short, is a really big deal. The World Health Organization even put it on a top priority list called escape pathogens. That's basically the sixth most wanted, most dangerous antibiotic-resistant bacteria out there. Now, what's crazy is that it lives totally harmlessly on the skin or in the nose of something like 60% of us right now. But if it gets into the wrong place, it can cause some really devastating infections.
Its real superpower, though, is its ability to build something called a biofilm. And a biofilm is pretty much what it sounds like. It's a fortress of slime that the bacteria build for themselves. This slime armor is made of proteins and sugars, and it makes the bacteria incredibly resistant to just about everything we can throw it at it. Our immune system, our best antibiotics, you name it.
So the scientists behind this new research noticed something kind of strange about how this slime armor was built. And that led them to a mysterious figure who seems to be running the whole construction site. And this right here, this is the key discovery that kicked the whole thing off. On the left, you're looking at a normal, healthy biofilm. It's a dense, thick carpet of bacteria. But look over at the right. This is what happens when scientists turn off just one single gene. The entire fortress just falls apart. It becomes all sparse and weak.
This, of course, led to the one massive question that drove their whole investigation. What on earth is this one gene doing that's so critical for the strength of the entire structure?
Well, the gene they turned off makes an enzyme called Nitric Oxide Synthase, or NOS. To make things simple, let's just think of this enzyme as the construction foreman for the biofilm. When the foreman is on the job, the fortress is strong. When the foreman's gone, the whole project just collapses.
And here's the hard data to back that up. The bar on the left shows the biofilm made by the normal bacteria. Business as usual. The bar on the right? That's what happens when you take away the foreman, NOS. Biofilm formation doesn't just dip a little bit, it absolutely plummets. It's a dramatic, total collapse.
Okay, so the foreman is gone and the fortress is weak. The next logical step for the researchers was to figure out what exactly the foreman was in charge of. What's the key material that was suddenly missing?
It turns out that the key ingredient, the mortar holding all the bricks of this slime armor together, is a super sticky sugar molecule called icaA. You can think of this as the superglue of the biofilm. It's what makes everything stick. And yup, the data confirms it. On the left, with the foreman on duty, the bacteria are pumping out tons of this icaA superglue. But on the right, when the foreman is gone, production of the glue just tanks. So the takeaway is clear, the foreman controls the superglue supply. But here's where the story gets a really fascinating twist. The foreman, this NOS enzyme, isn't actually making the glue itself. It's just giving the orders. The researchers found a middleman in the chain of command. The NOS foreman gives its orders to another protein called MgrA. We can think of MgrA as the molecular master switch that's directly in charge of the glue-making machinery. And this is where the puzzle got really deep. When the scientists got rid of the foreman, NOS, they checked on the master switch, MgrA, and they found that the amount of the master switch didn't change at all, but its ability to actually do its job, to bind to DNA and turn on the glue factory, became incredibly weak. So, what gives? If the foreman isn't making more of the master switch, how is it telling it to work so much better? There has to be some kind of secret signal being passed between them, right? And that question led to the ultimate aha moment of the entire study, the discovery of that secret signal. This signal is a tiny chemical modification called S-nitrosylation. It sounds complicated, but here's the gist. The NOS foreman produces nitric oxide, or NO. This little NO molecule then gets attached directly to the master switch protein, MgrA. And this simple chemical tag acts like a turbo boost, flipping the master switch into overdrive and supercharging its ability to work. So here it is, the entire elegant chain of command all laid out. 1. The foreman. NOS makes nitric oxide. 2, that NO acts as a chemical signal to the master switch, MgrA. 3, this signal, this S-nitrosylation, supercharges MgrA. 4, that supercharged switch binds powerfully to the DNA. 5, this cranks up the production of the icaA superglue. And the result? A strong, impenetrable slime armor.
And here it is, the whole shebang, the complete scientific blueprint. You can literally follow the process, from the NOS enzyme producing that NO signal, all the way to that signal modifying MGA, which then activates the genes to produce the PIS superglue that builds the fortress. It's a beautiful, precise little mechanism.
Now, figuring out this blueprint as more than just a cool piece of biology, it actually opens up a brand new way to think about fighting these superbugs. So why does all this matter so much? Well, for a few huge reasons. First, this whole system is found across all sorts of different strains of S. aureus, including the scary antibiotic-resistant superbug versions. This discovery gives us a totally new target. Instead of trying to just kill the bacteria outright, which they're getting really good at surviving, we can target this foreman. We can disarm their fortress and make them vulnerable again. And hey, don't just take my word for it. The researchers themselves suggest this could be a promising new strategy. Develop drugs that block the NOS foreman, and then combine those with our existing antibiotics. In other words, you take down the shield and suddenly the old weapons might just work again. And that leaves us with a really hopeful final thought. We now have the blueprint for the superbug's armor. So what if our next great weapon isn't a bigger, stronger antibiotic, but a simple drug that just fires the construction foreman? It could be a total game changer in the fight against one of our oldest enemies.